Hello everyone, my name is Amrita Sanjay Bhusle and today I'll be talking about protein, its structure, function and analytical techniques. Before going in depth about what proteins are, let us know what biomolecules are. So biomolecules are chemicals which are synthesized, utilized or interact with organisms. These molecules are essential for life processes like energy production, genetic information transfer, metabolic activities and facilitating various other biological functions. Biomolecules can be divided into inorganic biomolecules and organic biomolecules. Inorganic biomolecules are those which are not synthesized by living organisms, whereas organic molecules are those which are synthesized by living organisms. Now, why are we even talking about protein? How much relevant is it? Proteins are of extreme relevance because they are involved in multiple cellular activities as well as metabolic activities as listed over here. Now, what exactly protein is made up of? Protein is not a single unit. It is made up of amino acids. In fact, amino acids are of various types, but upon hydrolysis of protein, alpha amino acids are obtained. Now, what is the structure of an alpha amino acid? Alpha amino acid consists of a carboxyl group, hydrogen, amino group, variable group R, and a carbon atom. And when the amino group and carboxyl group are attached to the same carbon atom, we find alpha amino acids. There are 20 alpha amino acids and these are listed over here. While there are multiple ways of classification of alpha amino acid, one of the major ways is on the basis of R group. On that we have polar amino acids which are hydrophilic in nature, non-polar amino acids which are hydrophobic in nature, acid amino acid which are negatively charged at physiological pH, basic amino acid which are positively charged at physiological pH. Then we have aliphatic amino acid which consists of an alkyl group, aromatic amino acid which consists of an aromatic ring, sulfur containing amino acid which consists of sulfur. Moving forward. Now, as I said before that protein is not a single unit, it is a polymer of amino acids. What joins two amino acids together? It's the peptide bond. Peptide bond is an amide bond which is formed between a carboxyl group of one amino acid and amino group of another amino acid. How is it formed? It is formed by the process of condensation or dehydration in which a water molecule is also released. Now, what is the structure of protein? Well, protein structure is extremely complex and can be described at 4 levels. We have primary, secondary, tertiary and quaternary. The primary structure involves sequence of specific amino acids. The secondary structure involves two structures. One is alpha helix and one is beta sheets. Basically, there is infoldings or backfoldings at the polypeptide chain. Alpha helix is spiral in shape and beta sheets are pleated in shape. Then we have the tertiary structure which is basically overall 3D structure of a single polypeptide chain. While the quaternary structure is basically multiple polypeptide chain in a single unit. Moving forward, what is the function of protein? Well, protein can be enzymes, which is basically catalysts and can speed up chemical reaction. Most of the enzymes are made up of protein. For example, we have amylase which can break down starch into simpler sugars. Then we have protein as transport molecules which can transport other molecules like hemoglobin which can provide oxygen to tissues. Energy source. While human body's first choice is not protein for energy source, but then during intense exercise and fasting, protein can be used as an energy source. Then we have building blocks. Protein is used to build many other material as well. Then we have regulation. Proteins are involved in regulating gene expression. Example, we have cyclin which is responsible for regulating cell cycle. Hormones which are chemical messengers are also proteins. Example, insulin which is needed to regulate blood sugar. Then we have defense in which involves immunoglobulin which is an antibody which can fight against foreign invaders. Well, there are many ways in which we can study about proteins, but one of the ways is by studying through separation. Now, separation can occur by many techniques. In that also, we'll be talking about two techniques. One is chromatography and other is SDS-PAGE. Chromatography is a simple technique which is used to separate, identify and quantify components of a mixture. Working principle is also pretty much simple. It includes partition coefficient. What is it? It is basically concentration of phase 1 over phase 2. What are these phases? Phase 1 is basically a stationary phase which could be solid or gel or liquid. Then we have the mobile phase which is liquid or gas. What happens is when mobile phase moves over the stationary phase, particles get separated due to adsorption at stationary phase and differences in the partition coefficient. For protein, we have cation exchange chromatography where positively charged proteins can be separated. Then we have size exchange chromatography in which large molecules are eluted first and then small molecules are eluted. Then we have affinity chromatography in which proteins have an affinity to a specific chemical group through which we can separate them. And then we have HPLC, which is high pressure liquid chromatography in which high quality chromatography can take place under high pressure. Last technique which we are going to discuss is about SDS-PAGE. The second technique is SDS-PAGE, which is sodium dodecyl sulfate polyacrylamide gel electrophoresis, in which we are going to separate proteins on the basis of their molecular weight. Now SDS, which is sodium dodecyl sulfate, it is a detergent which binds to the protein, giving them a uniform charge to mass ratio. They also give negative charge to protein. So when electrophoresis goes on, proteins will move towards anode. Now migration occurs on the basis of size as said earlier, and smaller particles will move faster while larger particles will move slow. Now the gel we use is a polyacrylamide gel. It is a matrix at which migration will occur, and then we have electric field which will drive the migration. Now that polyacrylamide gel is further divided into stacking gel and resolving gel. Both of them have different pH because of the work of glycine, which is a zwitterionic molecule. The stacking gel's primary role is to concentrate the protein samples into thin, sharp bands before they enter resolving gel. This ensures that all proteins in sample enter the resolving gel at the same time. Glycine and chloride ions play a role in this. Then we have resolving gel, which basically separates them on the basis of molecular weight. After electrophoresis, proteins are visualized by adding dyes such as Kumasi brilliant blue or silver staining, which binds to proteins but not to the gel itself. SDS-PAGE is suitable for protein because it results visibility of protein with separation along with the determination of isoelectric point and approximate molecular weight. Thank you.